Ready to say?
Ready?
Okay.
Okay, Dr. David, we can start the presentation now. I just make some little presentation before you start. It is okay?
It's okay. It's okay.
Okay. Yeah, hello everyone. You are welcome in Molecular Genetics and Biotechnology Seminar Series, which organized by Department of Genetics, Faculty of Agriculture, Asyut University, Egypt, under the patronage of Professor Ahmed Al-Munshawi, President of Asyut University, Professor Jamal Badr, Vice President for Graduate Studies and Research, Asyut University, Professor Adil Wannan, Dean of Faculty of Agriculture, Asyut University, Professor Muhammad Al-Sughayar, Vice Dean for Graduate Studies and Research, Faculty of Agriculture, Dr. Ahmed Salam, Associate Professor of Genetics, Genetics Department, Asyut University. I am Dr. Said Mohammed. I am Associate Professor of Genetics at Genetics Department, Asyut University. I am the moderator for this seminar today. Our speaker for today is Dr. David Nobodo, with a PhD in pharmaceutical microbiology and biotechnology. Dr. David Nobodo has dedicated his career to unveiling the mysteries of microbial natural products and exploring their diverse applications in medicine, agriculture, and industry, with a keen interest in combating antimicrobial drug resistance. Currently, Dr. David is a MIF postdoctoral researcher at the Department of Pharmaceutical Science, University of Shizuoka, Japan. His academic contribution include over 25 peer-reviewed articles, book chapters, and several conference presentations, reflecting his commitment to advancing scientific knowledge. He participated as a peer reviewer in several prestigious journals. In addition, Dr. Nobodo is the founder and the editor-in-chief of Microbial Odyssey, a science blog that serves as a beacon of knowledge about microorganisms, microbiology, and biotechnology. Dr. David, a presentation today entitled Biosynthesis of E-beta-Francine and 1-bipyronyl 3,5-diamino-1,2,4-triazole through synergistic coculture of endophytic L. theobromine and C. lunata. Dr. David has 20 to 30 minutes for his presentation and another 15 minutes to answer our question. Dr. David, please, you can start your presentation now.
Okay. Good evening, everyone. So thank you very much for the introduction. So my name is Dr. David Nwobodo from Nigeria, a researcher and a lecturer with the Renaissance University in Enugu, Nigeria. Currently on my postdoctoral fellowship at the University of Shizuoka in Japan. I want to thank the organizing committee, Department of Genetics, Asyut University, Egypt, for accepting my abstract and giving me the opportunity to make this presentation. I also want to thank Mr. Abdullah Rafiq, who contacted me initially and made this possible. I'm happy to be here and I'm happy to be making this presentation. So without wasting much time, I will go straight to the presentation talking on the biosynthesis of β-Farnesene and 1-bipyronyl 3,5-diaminotriazole through the synergistic co-culture of endophytic Laciniopludia tuberome and Covularia lunata. My research has generally focused on the discovery or the search of natural bioactive compounds from natural sources, which could be applied in pharmaceutical, agricultural, and industrial sectors. And these natural products actually can be gotten from different sources, such as the plants and the microorganisms. But my focus is particularly on microorganisms, the group fungi, and the group of organisms known as the endophytes. Endophytes are basically organisms that reside or live within plant tissue in a commensal relationship without causing any harm to the host. Rather, some of them have been shown to help the hosts withstand hostile environmental conditions such as pests and droughts by the production of secondary metabolites. So these organisms are known to produce different, diverse, complex secondary bioactive metabolites, which can be applicable in the aforementioned sectors. However, in our search for these natural products, science is gradually drifting from the traditional technique that has to do with just isolating these organisms in the laboratory and then just allowing them to produce these compounds, to moving to the level where we have to engineer these organisms by influencing the regulatory systems that govern the expressions of these secondary metabolites. That is, engineering these organisms to be able to see how to activate or induce these genes that are responsible for secondary metabolites production in the laboratory, with the aim to enhance the productions of these compounds and their isolation. And one technique that is visible or that have stood out recently in this regard is the co-culture technique, which has been identified as a powerful tool for enhancing the expression of weakly expressed biosynthetic pathways in microorganisms in the laboratory. So why is this research important? In Nigeria, in Africa, in Asia, and a large population of the world rely heavily on medicinal plants as their source of therapeutic agents in traditional medicine and complementary medicine. So this has led to or could lead to heavy deforestation, heavy plant harvest, leading to ecological imbalance. And then there comes endophytes, which we think would serve as a worthy alternative to these plants. And why we think this is a worthy alternative is because, one, endophytes have been shown or have been reported to produce similar or even better compounds than their host plants. And then secondly, with the ability to isolate these endophytes and then engineer them, we have the opportunity to produce even better compounds in the laboratory, novel compounds that were not produced by the host plants. And so, which is why most times in the isolation of endophytes or in the exploration of endophytes for the production of bioactive compounds, we tend to look for endophytes from medicinal plants, because we know that some hypotheses also claim that these endophytes are responsible for the production of these compounds by their host. However, there is the biosynthetic pathway that are responsible for the secondary metabolite production in these microorganisms are controlled by biosynthetic gene clusters. But the expressions of these gene clusters remain cryptic or remain idle or latent under laboratory conditions. So when these organisms are isolated and brought to the laboratory, they are not able to fully express this. So what we look at is how do we make these organisms to be able to activate or enhance the production of these compounds by activation of these cryptic or weakly expressed gene in the laboratory. So that is what we try to do with co-culture experiments. So just two objectives. First, to investigate the biosynthesis of bioactive compounds by using the co-culture experiments of two fungi, Lactobacillus tuberosum and Corbillera lunata. And then we carried out comparative analysis of the metabolic extracts from the monoculture, which is the individual cultures, and the co-culture using chromatographic techniques. So here is just a summary of this methodology adopted in this study. First, we selected plants Cola acuminata, and Elis guinensis, from which the plant leaves were collected and treated, cultured on malt extract agar for about 6 days. Then we started seeing growth. From these growths, the organisms were then purified. So these are the pure isolates, which were now used individually for fermentation and then combined together to carry out the co-culture experiments. Both the monoculture and the co-culture were cultivated or fermented on localized medium for 21 days after which the crude extract was collected by ethyl acetate solvent and concentrated, then tested for biological activity just to see the difference between what is happening in the crude extract of the monoculture and the co-culture. Then we use GCMS, and then I think HPLC and GCMS to check for the chemical profile of the crude extracts obtained. So L. tuberoma and Silonata were isolated from Cola acuminata and Bambusa vagularis, the leaves of these plants respectively. These organisms were identified molecularly by sequencing the ITC, internal transcriptional region of the fungi, which was submitted to the blast to the NCBI website and returned with the following accession numbers currently submitted to the gene bank. Then L. tuberoma has been isolated severally from different plants, same as C. lunata, have also been isolated as common endophytes associated with different plants in time past. So this is a pure culture of the L. tuberoma and pure culture of the C. lunata. So for this presentation, we only ran the antibacterial activity just to look at the difference. So we saw that at 1 milligram per ml concentration of the extract from these organisms, the monoculture, L. tuberoma, C. lunata, and then when combined in the experiments, we saw that L. tuberoma was only able to inhibit Staphylococcus aureus, while C. lunata extract inhibited Staphylococcus aureus, Bacillus subtilis, and Pseudomonas aeruginosa. When both organisms were co-cultured and then crude extract tested, we found a slight increase in the concentration in the inhibition zone against Staphylococcus aureus, but there was no other inhibition against any other test bacteria isolates. Even the Bacillus and the Pseudomonas that was originally inhibited by C. lunata, the ability was lost. So this tells us that some compounds have been antagonized, while some compounds that are responsible for inhibiting Staphylococcus aureus were actually enhanced in the co-culture extracts. So here there's a clear zones of inhibition obtained in the antibacterial analysis. So we looked at the chemical profile of the crude extracts, and here is an overlay of the chromatogram obtained in the monoculture and the co-culture. The red curve is for C. lunata, blue L. tuberoma, and green is the curve for the co-culture extract. We see that compound A was identified in the 3 extracts used. However, we see that the peak on the co-culture extract, there was a reduction in the peak size or peak height in the co-culture as compared to the monocultures, while peak B was absent in the co-culture but present in the monocultures, while on the other hand, peak C and peak D were not present in any, so exclusively produced by or peaked at the co-culture extract. So this means that some compounds were actually produced. Some new compounds like C and D were actually produced in the co-culture experiments. But we were not able to really identify these compounds because the HPLC we had was not attached to mass spec. So we decided to run GCMS analysis to see if we could be able to identify these compounds. And out of the 13 compounds, that's unique 13 compounds that were produced or identified in the co-culture extracts, 49% of them, 8 were new compounds. That is, compounds that were not produced or not identified in the extract of L. tuberoma or C. lunata, the monocultures, while 10%, about 7 of them, were antagonized. 7 of them were antagonized. So this is just a table showing a highlight of the most important, the most abundant compounds that were identified in the crude extracts. So we observed that compound 6, β-Farnesene, compound 10, beta-sesquiphyladrine, and 13, 1-papironate-5-diaminotriazole, were identified at very high concentrations when compared to other components or other compounds in the extracts. So endophytic fungi have previously been isolated from Cola acuminata and Aedes guinensis. However, our study was the first to report the isolation of both organisms from these two plants, Cola acuminata and Aedes guinensis. There are reports that close microbial interaction trigger biosynthetic pathways in microorganisms, and our findings support this hypothesis that ecological context influence microbial metabolite production. The co-culture resulted in the production of reasonable concentration of compound 6, compound 10, and compound 13. Compound 6, 10, and 13. And statistically, the concentration of compound 6 and when compared, concentration of compound 6 and compound 12 were significant, scientifically significant when compared to the concentration of all other compounds identified in the co-culture extract, which means that these compound 6 and compound 13 were not identified by chance, rather as a result of the interaction between the two organisms used in the co-culture experiment. So let's quickly look at the biological activities and applications of this. So β-Farnesene is a secondary metabolite that is known to be produced by several plants and microorganisms alike, and they serve as signaling molecules in plants' defense mechanisms. That is, they are released in response to herbivore attack to chase pests away. So they have been reported as good components for the formulation of biopesticides that are environmentally safe and friendly. On the other hand, they've been reported also to be used in flavor and fragrance industry to enhance the flavor of food products, cosmetics, and fragrances. So this study shows a natural source of this compound that can be used for this particular purpose. Then secondly, 1-papironate-5-diamino-1,3,4-triazole have also been shown to be a potential compound that can be applied in medicinal chemistry, organic synthesis, and pharmacology. And this is basically due to the structure of this compound, the presence of pypronium moiety and the triazole ring, which is found in this compound. And previously it has been reported that the 1,3,5-diaminotriazole nucleus is a potential scaffold for the design or development of anti-cancer agents. So as a result, this compound could serve as a foundation for the development of newer anti-cancer agents. So mostly the pypronium derivative of this compound is scarce, like there are no limited information about this, which shows that it could serve as a good starting point, like I said, in the development of anti-cancer, new or anti-cancer drugs. So what is the application of our studies? The results here validate claim that fungi endophytes are excellent reservoirs of bioactive secondary metabolites. Co-culture has shown to be an effective strategy in producing new bioactive compounds not produced by individual cultures. And the findings provide proper framework for utilizing co-cultivation as a technique for activating cryptic genes and enhancing bioactive compound synthesis in fungi. So I recommend that the biosynthetic pathways of these organisms that produce these natural products should be properly studied so that it can be able to lead to scale up and industrial applications, then expansion to other fungi species for novel drug discovery. So basically, currently, this first recommendation is what I'm currently doing now in my post-doctoral research, where I look at the biosynthetic pathway of an anti-cancer natural product, and then see how we can scale it up, not just the production, but to see how to make cells to be able to, human cells to be able to produce these compounds. So my final thoughts, after this research and going through several literature, I came to the conclusion that although microbial co-cultivation have proven successful in inducing secondary metabolite biosynthesis, it is still an arbitrary approach that suffers from low predictability. So it is very low for you to be able to predict the outcome of co-culture experiments, because you don't really know which of the organism is the elicitor, which of the organism is responsible for the production of the new compound, or which organism, which gene, or which gene cluster in which organism is responsible for this. So more study is required to be able to understand the two organisms involved in co-culture experiments, to be able to have a fruitful use of this technique in drug discovery. So selected references. Thank you very much for listening. I await questions.
Thank you, Dr. David. For the nice presentation. And we have 15 minutes for the audience question. If anyone has question, you can put their hand.
Please can you mute your laptop and switch one device, your mobile or lap, to find error in your mic.
I thank Dr. David for your nice presentation. For the audience, if anyone please has a question, he can push the raised hand on the screen, or he can write his comment on the chat box. Anyone of the audience has a question?
I think Dr. Joseph has question.
Yes, I have a question. Yeah, good evening, all. Can you hear me?
Yeah, hello.
Good evening, all. This is Ike Joseph. I want to commend Dr. David Wobodo for a wonderful presentation. And I have I want a clarification from him. My question goes on the methodology, on the extraction of the endophytic fungi. Please, I want to clarify, how is he sure it wasn't an epiphyte that was isolated?
Can I respond?
David? Okay, you can respond. You can start responding.
Okay. So during the process, the plant leaves, when harvested, they are treated first using four steps. First, washed in running tap water, and then treated on 1.5% hypochlorite, then 70% ethanol, and final wash in water before being cultured on malt extract agar. And the malt extract agar is being fortified with about 50 milligram chloramphenicol to prevent the growth of bacteria, and then finally, to be sure that the growth of the endophytes, the organism that is growing, the fungi growing here, are only endophytes, you run a control where after the treatment, the plant leaves or the midribs are placed on the medium and then removed. They are pressed on the medium sideways, front and back on the medium, and then removed, and then culture alongside this plate. So if there is a growth after 7 days or thereabouts on the medium, then if there is a growth before or within the space of 7 days on the medium, the control medium, that means your treatment process was wrong. But if there was no growth on the control medium, that means the treatment process was correct, and that all organisms growing are endophytic organisms from the plant. I don't know if that answered the question.
Yeah, thank you very much. Thank you very much.
Thank you.
Thank you. I think we have another question from Hinodigo. Yeah, we have another question from Hinodigo. Dr.
Yuan, can you unmute your mic?
Okay. Can you hear me now?
Yes.
Hello?
Yeah, you can start your question.
Okay, I can't. What's that? Can you hear me?
Yes, we can hear you.
Thank you. Mike, thank you very much for your interesting presentation. I have a couple of questions. The first one is, in the way you identify the endophytes, whether they have an effect or not, is treating them alone and with gentamicin. But I didn't to compare the effect of gentamicin compared to the endophytes. Why you haven't done a separate gentamicin test on the on your experiment? Then we can see clearly the effect of the endophyte isolate compared to gentamicin. So the second question is, in your HPLS analysis, I was not able to understand how you define significance peak in the HPLS, because some of the low curves are there, but you mentioned it as if it is it is a curve which indicates some kind of chemical or the metabolite. So is there any standardization to say the HPLS above this level of curve is really significant, or below that is not significant? If you explain that for me, thank you very much.
So for the first question, I didn't get it too clearly. You said, can I separate gentamicin? Is it here? Is it this table?
Yes, on that table, I can see that you have the test isolates, and
okay,
you have the different, here, for example, 12 alone with theobromine, 8 with lata, and when you are mixing it, it becomes 14. And you added gentamicin to it, it increased to 20, right? So you know that gentamicin is a well-known antimicrobial activity and strong one.
Okay.
So
okay,
if you have a separate one alone with this isolates. Thank you.
Okay, let me explain the table clearly. So what we have here is not combination. So after this has been obtained, we fermented this. After fermenting this, we got the extracts of this, extract of this, extract of this, and extract of this combined. extract of this, and extract of this combined. So that was what we represent here. So this is the extract of this alone tested against this. Crude extract tested against this. Extract of this alone tested against this. Extract of this alone tested against this. This served as the standard positive control. It is not a mix. It's not a mix, a combination. I don't know if I got it right.
Thank you. It's clear now.
Okay.
And about the HPLC, yeah. So if I see here, there are like a strong curve with the red one and the blue, but while in the green, it is really small. But you pointed about that. That like the first blue curve is what is it? A control or what's that? The green curve.
So the green curve is the curve. So these are different curves, separate curves for the different treatments. The red one is, like I explained in the antibacterial as well. The red one is the curve for the extracts alone for C. lunata. The blue one is the curve for L-Tribome, and this is the curve gotten from the extract of L-Tribome and C. lunata. So what happened was that in order to be able to understand, because like I initially said, the major limitation we had in this particular chemical analysis is that the HPLC that we use for this study wasn't attached initially. It wasn't attached to a mass spec. So we only saw curves. So to make a meaning out of the curves that we saw, we tried to compare side by side the different extracts to look at matching curves. So we can say, okay, if this curve, like I try to say in this A now, the curve on a straight scale, on a straight line, we could see that there is a peak at the same point for this compound, this particular peak. While there is a difference, it is different here. So there's no peak on the green here. So it just tries to give us, we cannot categorically say that this is a compound or this is not a compound. So what this chromatogram just shows us is that, okay, even though we're not able to really identify these compounds or to say that this compound is this or that, but it shows us that there is a change, there's a pattern of change in the extracts of these fungi, which correlates with something that we found there. So we see that here, there is an increase when these two organisms are used together and then produce a compound. There's an increase a bit, while there is a decrease in this against this organism, because it did not inhibit this organism that was inhibited by this alone. Same applies to Pseudomonas aeruginosa. So we found that there is a decrease or a complete loss in the compound that inhibits this and this. So, and the HPLC that we got is showing us a similar pattern. Yes, we cannot be able to categorically say that, okay, we are sure that this, which was why we now went for the GCMS analysis. So I don't know if this makes any sense.
Very good. Yeah, thank you.
Thank you very much.
Thank you, Dr. Iwan. Anyone has another question or any question? Anyone has a question? Abdullah Rafiq has a question?
Yes, I have a question.
Or Abdullah?
Dr. David, I want to confirm my understanding of the use of coculture. Did you combine the extract of the two strains in the plate and place them in one conical contain rice medium?
Please come again. Sorry, I didn't get you clear.
I want to confirm my understanding the use of coculture. did you combine the extracts of the two strains of fungi, place them in rice media? You
Yeah, so
understand?
what happened was that we, like, after the pure isolates have been obtained, this alone cultured, take for instance, this alone cultured on this particular conical flask alone, this cultured on this alone, and these two cultured in this combined, and this.
Okay.
So it's not the extract that are combined, it's the organisms itself, the mold itself, 1 and 2 cultured in this and this cultured in this cultured in this, and these two combined together cultured in this and this. It's not the extra that is combined. So you allow them to grow. During this process, you allow them to grow to not to full confluency like this, like 100% or 90%. When they are still very young, maybe say at a little age, around circle like this, not when they have gotten to this level. You combine them and they keep.
No antagonistic with others. Okay, right?
You said?
No antagonistic with other.
Sorry, I didn't get you. No genetic?
Antagonistic with them.
Antagonistic?
Yeah.
Did you say no antagonistic? Yeah, the antagonistic. We observe some antagonistic reactions, such as reducing. So a situation like, take for instance, this, we observe that, okay, take for instance, this now, this compound number 2, these two individual compounds produce high concentrations of these compounds, even though it's just a straight hydrocarbon, high concentrations of this compound. But when combined, there was an antagonism. The concentration reduced drastically. So, and we believe that same thing happened in this, where this was produced by this alone, but when combined, there was a reduction. I don't know if that is what you mean. So there are synergistic, but I'm more interested in the outcome of the synergistic, because they gave us very high concentration of compounds not produced by any of these. And again, the compounds that were produced as a result of the synergistic outcome were actually good compounds that are currently needed for applications. So we didn't really look at the antagonist, but I highlighted that some compounds here were actually antagonized.
Okay.
I don't know if I got you correctly.
Okay. Thanks, Dr. David.
Thank you.
Thank you, Dr. David.
I have other question, Dr. Said, please.
Okay.
Yeah, you can ask.
Could you provide information regarding the quantity of your two strains necessary to yield a specified amount of its constituent components?
Specify the amount of what components, sorry?
Your components, two components. I don't remember the name of components you explained. Two components extract from two fungi.
You mean this?
Francine, I think that Francine and
Okay. This, you mean this?
Yeah, yes. Beta.
If I can,
yes.
If I can provide
Beta.
the components.
Yes.
Hello,
Dr. David. Could you provide information about the quantity of two fungi to yield the specified amount of these components?
Wow, sorry, I didn't get this question very well. I didn't get it.
I think this further studies, but I think you have any information about that.
Can you specify your question, Abdullah? Abdullah, if you may specify your question, you can make your question is more clear or specific. Abdullah, you can hear me?
I hear you, Dr. Said.
Yeah, I mean, you
My
can
question
make
is
your
clear or not
question
clear?
is more clear and more specific.
Okay. I want to ask about the quantity of strain of fungi to get the specified amount of this component.
You may your question have the amount of each strain in the culture.
So you asked the quantity of these organisms that yielded this quantity of these compounds identified, right?
Yes.
Okay, so
I mean, like CFU of bacteria.
So we didn't take into consideration the quantity because what we did was 2 plates of this,
two
plates of this, and 2 plates of this.
Okay.
Cultured in, I think 200 grams of rice, local rice medium, for 21 days. So that was just there was no extra metric that we used in calculating the quantity of this that gave us that. So this was like a pilot study.
Yeah, it's a pilot study. Yeah, I can.
So if you notice, that's why I try to say, because much is not really known. That's why I made this final thought that most times predictability is difficult when doing co-culture. So for us to be able to have more predictable outcomes and more direct, because sometimes after this you find out that when you co-culture these 2 organisms again, there are chances that these compounds might not be produced. But why this stood out is that the concentrations of these compounds, these concentrations appear to be significant enough, very significant enough compared to other compounds that were identified in this thing. So what we did was just we combined 2 plates of this, 2 plates of this in 1 conical flask, 2 plates of this, 2 plates of this in 1 conical flask to be able to get enough yield for analysis. Because initially this study is just part of the study. The extract gotten from this were used for various other biological analysis, not just this and then HPLC. So we checked for a lot of other components like antiplasmodial activity, that is anti-malaria. We checked for antioxidants. We checked for antifungal. We checked for several activity bioassay analysis. But for this presentation, I only presented this, not to make the whole thing complex.
I think you got your question, your answer, Abdullah.
Thanks, Dr. David and Dr. Said.
Thank you, Dr. David. I think question, and I'm sorry about that. You got many questions today, but my question is just a simple question: is the mechanism why the coculture has a different, can give us different product when we use one species culture. If you can make a culture by one species, and you can make a culture, co-culture one, the product is different. It is well-defined mechanism.
Sorry, you said, why is it different that when you culture?
I mean, when we make a co-culture, when we can make a co-culture, we can find the different quantity of the product when we compare using making a culture using one species of organism.
You said when we make co-culture?
Yeah, when we make a co-culture,
Yes.
the product of co-culture is different compared to using one species when we make culture for one species.
Are you asking why?
Yeah, when we make a culture, you have this type of culture, co-culture, and then a culture using one organism and another culture with different organisms, right?
Yeah.
You have three type of culture. Okay, one of them is co-culture. Another culture, you have used only one organism inside each culture, right?
Yes.
Yeah, so I mean the product, when you separate the product for each culture of them, you got different quantity of the products, right?
Yes.
Like a Fransene or... So is there a well-defined mechanism can explain why the co-culture give us a different result?
Okay, so basically, like I said, if organisms tend to interact, the interaction between organisms lead to several, like this second point, I say close microbial interaction trigger biostatic pathways. So in ecology, most organisms, when they come together, the interaction, just like what happens in quorum sensing, there is an interaction between these two organisms or several. Remember, in an ideal situation, say in plant leaves, in human body, and then in the environment, organisms do not live in isolation. So that is why most times when we try to look at getting these organisms, sometimes when you isolate these organisms and bring them to the laboratory, they're not able to produce the compounds that they naturally produce in their host cell because of some interactions between organisms lead to the activation of some certain genes in those organisms responsible for that production. So it could be that when these two organisms were co-cultured together, either as a result of competition with nutrients or either as a result of one chemical signal or the other, they are able to activate certain gene clusters that ordinarily in isolation these organisms will not produce, leading to the production of different other compounds. And this basically tries to explain why when you bring two organisms in a co-culture experiment, you see different compounds from when these organisms are grown separately. So I don't know if this makes any sense.
Yeah, okay. Yeah, thank you. Yes, thank you for your massive presentation, and thank you for your explanation and interpretation of, for our question. And thank you for everyone, and we hope to see you after the next week.
We have questions in chat room. I think that
Who has anyone has another question?
in chat room we have questions. Do you first identify the bacteria and what methods you used for this?
Identification of the bacteria or the fungi?
Bacteria.
Which?
Bacteria.
Bacteria?
Yes. You first identified the bacteria and what methods you used for this?
Which bacteria? You mean this bacteria?
Yes, Dr. David. Yes.
Okay. So these were laboratory isolates using Begai's standard, they are already stock cultures from our laboratory. These isolates that were used for the antibacterial analysis. So, but for the fungi, we use molecular techniques by lactophenol cotton blue first, and then check for the morphological characteristics of the organism. And then in addition, we now sequence the ITS region, the internal transcribal spatial region of the fungi, and then gene blast. And the sequences are currently in the gene bank, NCBI gene bank.
Okay, Abdullah.
Everything is good, Dr. Sayed.
Abdullah. Abdullah.
Okay, Dr. Sayed.
Abdullah.
I hear you.
Abdullah.
Dr. Sayed, I hear you. I hear you.
Abdullah.
Yes, Dr. Sayed, yes.
Abdullah.
Dr. Sayed, I hear you.
Dr. Sayed. Yes.
No.
You got
No,
your
Dr.
question?
Sayed. Yes. No question.
Abdullah.
Yes, Dr. Sayed. No question.
We have no more questions.
I think Dr. Sayed have favor in your mind. Dr. Sayed has problem in your mind. Please, Dr. Sayed, mute your device. One device.
Yeah. Yes. Hello.
Yes, Dr. Sayed, I hear you. I hear you, Dr. Sayed.
Yeah.
I think Dr. Sayed has a problem in your mic. I think Dr. Sayed has problem, so I will end with you, Dr. David, and thank you for your informative lecture, and see you again in the next seminars.
Thank you very much. I appreciate it.